We have already discussed about the pre-fertilization events that occur in stamen. Now we will focus on the pre-fertilization events and structures in pistil, megasporangium, which is also called ovule. Here we will also discuss the development of embryo sac, which is also called female gametophyte. It is called female gametophyte because it will contain female gamete, that is egg. Parts of pistil or carpel, as you all know, stigma, style and ovary. Stigma is the landing platform for the pollen grains. Style is the elongated part next to stigma, and ovary is the basal bulged part of the pistil. Female reproductive part of a flower is called gynoecium. In a flower, there can be single pistil or carpel, then it will be called monocarpellary gynoecium. In some cases, there are more than one pistil or carpel. In that case, the gynoecium is called multicarpellary gynoecium. Multicarpellary gynoecium is of two types depending upon whether the pistils are fused or they are free. If the pistils are fused, you can see stigmas are also fused, then that type of gynoecium is called syncarpous gynoecium. And if the pistils or carpels are free, then the gynoecium is called apocarpous gynoecium. Example of syncarpous gynoecium: tomato, tulip, hibiscus, and that of apocarpous gynoecium: rose, lotus. If we look at a transverse section of ovary, in some cases we may find chambers in an ovary. These chambers are called locules. Here we can see three chambers, 1, 2 and 3. So this ovary is trilocular. And in each ovary are present ovule. Ovules are also called megasporangia. Ovules are attached to a special tissue called placenta. Ovules are attached to placenta through a stalk called funicle. In an ovary, number of ovules may vary in different plant species. In wheat, paddy and mango, there is only one ovule in each ovary. In papaya, watermelon, etc., there are many ovules in an ovary. Now, the structure of megasporangium or ovule. Here, the single ovule is shown in the ovary. Now, ovule is having two coverings, outer covering and inner covering. Outer covering is called outer integument and inner covering is called inner integument. These integuments are not continuous. These integuments are absent at a point and that point is called micropylar pole. And the pole which is opposite to the micropylar pole, that pole is called chalazal pole. Next to inner integument, there is cellular tissue called nucellus. And then next to nucellus is a 7-celled structure called embryo sac. This embryo sac has 3 cells at the chalazal pole and all the 3 cells are called antipodals. And at the micropylar end, there is a 3-celled egg apparatus. The center cell of the egg apparatus is called egg and surrounding this egg are 2 synergids. In the center of the embryo sac is a cell called central cell with 2 polar nuclei. Next is Megaspore Genesis, that is formation of megaspore from megaspore mother cell by meiosis. The site of megasporogenesis is ovule. Megasporogenesis occurs inside the ovule, or we can say it occurs in megasporangium. Now here a diagram is depicting ovary, ovule, ovule with both the integuments and only nucellus. Embryo sac has not developed yet. Only one cell of the nucellus at the micropylar end behaves as megaspore mother cell, and that megaspore mother cell will undergo meiosis to form megaspore, and from that megaspore will be embryo sac formed. Now we will discuss in detail the development of embryo sac or female gametophyte. One of the cell of the nucellus at the micropylar end increases in size and starts behaving as megaspore mother cell. This megaspore mother cell will undergo meiosis 1 to form 2 daughter cells. These daughter cells will undergo meiosis 2 to form 4 daughter cells called megaspores. These are called megaspore tetrad. Out of these, 3 megaspores will degenerate and only 1 megaspore is functional. And from that functional megaspore, embryo sac will develop. How? Let's see. That functional megaspore will undergo 3 successive mitotic divisions. Here, in this case, in this mitosis, nuclear division is occurring, but that nuclear division is not followed by cell wall formation, such that a single cell is having 2 nuclei. Again, when this cell will undergo meiosis, only nuclear division is occurring, cell wall formation not occurring. So, this cell is having 4 nuclei. Then again mitosis, now the cell is having 8 nuclei. Now these 8 nuclei, they will rearrange themselves and simultaneously cell wall formation occurs around them. 3 of these cells, they move towards chalazal end, and these 3 cells will then be called antipodal cells. 3 of the cells move to the micropylar end and constitute the egg apparatus, with center cell called egg surrounded by 2 synergids. These synergids, they have cellular thickenings at the micropylar end. These are called filiform apparatus. This filiform apparatus helps in guiding the pollen tube towards the synergids. In the center, there is central cell with 2 polar nuclei. Now this whole structure is embryo sac, which is also called female gametophyte because it is having the female gamete egg. So we have discussed from the megaspore mother cell, 1 embryo sac is formed. Here the embryo sac formation occurs from a single megaspore, so the development of embryo sac is also called monosporic development. Monosporic development of embryo sac means embryo sac has developed from a single megaspore. 3 megaspores degenerate. Only 1 functional megaspore forms the embryo sac. If we count the number of nuclei and the number of cells in an embryo sac, we will see that embryo sac is 8 nucleate and 7 celled. How? Let's count the number of cells in an embryo sac. There are 3 antipodal cells, 1 central cell, 2 synergids, 1 egg. So the total comes out to be 7 cells. Now if we count the number of nuclei, 3 antipodals, 3 nuclei, 1, 2, 3. 1 central cell, but here are 2 nuclei present. Here lies the difference. So one central cell with 2 nuclei. Then two synergids with 2 nuclei, one egg with 1 nuclei, and the total number of nuclei come out to be 8. So we can say embryo sac is 8 nucleate and 7 celled.